Hello everyone, welcome to my YouTube channel. I hope you all are fine. So this is the last video of chapter 1 BRS Physiology, and the topic is smooth muscle. So in the previous video we discussed skeletal muscle, its structure and the mechanism for excitation and contraction in skeletal muscle, and I will follow the same pattern for smooth muscle. So let's get started. Okay, so the first slide here, let's talk about the difference between the smooth muscle and the skeletal muscle and cardiac muscle. So unlike the other two types, the smooth muscles does not have sarcomeres, and we know that the sarcomeres are the contractile units that are found in the skeletal and cardiac muscles. But the smooth muscles, they do not have the sarcomeres, and because of this, they do not have the striated pattern that we have seen in the skeletal muscle, and which is why it is called smooth. However, the smooth muscle still contains the same thick and thin filaments, actin and myosin, present in the skeletal and cardiac muscles. And these filaments work together in the sliding filament mechanism to produce contraction. Okay, so it is important to note that there are actually two subtypes of smooth muscle tissue. One is the multi-unit smooth muscle, and the other is unitary or single unit smooth muscle. And single versus this multi refers to the number of nerve fibers required to activate the smooth muscle tissue. Okay, so the single unit smooth muscle or the unitary smooth muscle, it is innervated by only one or very few nerve fibers per bundle. Okay, so you can see here that the single unit smooth muscle, it has only one nerve fiber, and there is no need for many nerve fibers because one nerve fiber can contract the entire sheet of smooth muscle in union because of the presence of gap junctions. And these gap junctions, they allow the electrical signals to pass from one cell to the adjacent smooth muscle cell rapidly. So the single unit smooth muscle, it is present in the hollow organs like the intestine. So it is the most common type that is present in the uterus, the gastrointestinal tract, ureter and bladder, and that's why it is called as the visceral smooth muscle. And it is spontaneously active, exhibit the slow waves and exhibit the pacemaker activity, which is modulated by hormones and neurotransmitters. It has a high degree of electrical coupling between the cells and therefore permits the coordinated contraction of the organ. For example, bladder. So this is we talked about, that is the gap junctions, and because of the gap junctions they have the high degree of electrical coupling between the cells. Okay, so unlike the single unit smooth muscle, the multi-unit smooth muscle, they do not have the gap junctions, so each cell requires its own electrical impulse. Hence, there are multiple nerve fibers. So it has many nerve fibers and they do not have gap junctions. So because of a lack of connection between the cells in the multi-unit smooth muscle, each cell requires its own electrical impulse and it behaves as a separate motor unit. And the multi-unit smooth muscle is densely innervated. The contraction is controlled by the autonomic nervous system. Okay, then we have the excitation-contraction coupling in the smooth muscle. So remember that there is no troponin. So how the intracellular calcium increases? Okay, so we have three mechanisms. One is the depolarization of the membrane. It opens the voltage-gated calcium channels. And calcium flows into the cell down the electrochemical gradient, and this increases the intracellular calcium. And the second is the hormones and neurotransmitters. They open the ligand-gated calcium channels in the cell membrane. So by both these mechanisms, depolarization, which causes the opening of the voltage-gated calcium channels, the hormones and neurotransmitters, which open the ligand-gated calcium channels in the cell membrane, they will increase the intracellular calcium, and this causes the calcium-induced calcium release from the sarcoplasmic reticulum. Okay, so the third mechanism is also by the hormones or neurotransmitters, but now they directly release calcium from the sarcoplasmic reticulum by this mechanism, that is through this IP3 mechanism, inositol triphosphate gated calcium channels. And this causes the release of calcium from the sarcoplasmic reticulum directly. And by all these three mechanisms, we have this increase intracellular calcium. Okay, so when the intracellular calcium increases, calcium it binds to calmodulin, and we have a calcium calmodulin complex. The calcium calmodulin complex activates the myosin light chain kinase. When activated, the myosin light chain kinase phosphorylates myosin. So when we have the phosphorylation of myosin light chain, it allows it to bind to actin, and thus initiating the cross-bridge cycling, and this results in the contraction of the smooth muscle. Okay, so this means that the amount of tension that is produced in the smooth muscle, it is proportional to the intracellular calcium concentration. So what happens when there is a decrease in intracellular calcium? The muscle relaxes. Okay, and lastly we have this table from BRS Physiology, that is the comparison of skeletal, smooth and the cardiac muscle. Leave the cardiac muscle aside because we will talk about it in detail in the chapter 3. So the skeletal muscle and the smooth muscle, how they are different? They are different in appearance because the skeletal muscle, it is striated, and the smooth muscles, it has no striations. And this is because the smooth muscle do not have sarcomeres. The upstroke of action potential in the skeletal muscle, it is because of the inward sodium current. But in the smooth muscle, it is because of the inward calcium current. Okay, so plateau phase, it is only a speciality of the cardiac muscle. So the smooth muscle and skeletal muscle, they do not have this plateau phase in their action potential. And the duration of action potential for skeletal muscle, it is 1 millisecond. For smooth muscle, 10 milliseconds. Okay, so the excitation-contraction coupling in skeletal muscle, when the action potential, it reaches the muscle membrane, it causes a depolarization of T tubules. Calcium is released from the sarcoplasmic reticulum by the calcium release channels, and this results in the increased intracellular calcium. And what is the molecular basis for contraction? How the cross-bridge cycling it starts when the calcium binds to troponin C. And for smooth muscle, the excitation-contraction coupling, how it occurs when action potential, it opens the voltage-gated calcium channels in the cell membrane, or the hormones and neurotransmitters, they open the IP3 gated channels in the sarcoplasmic reticulum, or the ligand gated channels. So this is how the excitation-contraction coupling occurs. And what is the molecular basis for contraction in the smooth muscle? Calcium, because smooth muscle do not have the troponin C. So calcium binds to calmodulin, and this increases the myosin light chain kinase. Phosphorylation occurs, and this results in the cross-bridge cycling. That's simple. I hope everything is clear to you. So we have completed chapter 1 BRS Physiology. Thank you so much for watching. I'll see you all in the next video.